Hello, everybody, and thanks so much again for the invitation. And greatly honored to be a member of the great discussion that we're having. And I follow Dr. Murad with another targeted genetic alteration that is important for us in HCC. So I'm going to go ahead and share my screen. And what we're going to do is we'll be talking about FGF2 after what Dr. Brad mentioned in regard to the in regard to the IDH1. These are my disclosures, and these are my contacts. You can go ahead, please, and if you want to take a picture of the QR code, you can connect with me. And as everybody knows, I'm readily available at any time for helping out in any way I can. And with this said, we're going to focus on the FGFR, as we just mentioned, and we'll touch on a little bit on the basis of it. We'll talk about the phase 2 studies, and there's plenty of them. We'll talk about resistance. And of course, the question is, where do we go from there? In regard to FGFR, that's the simplistic way of really what it is, and it helps me, and hopefully it will help all of us to kind of understand better. It's a a transmembrane important protein that will through a spiral connection ultimately will connect to the RAF-MEK pathway that we're all familiar with, and everything happened right up there at where the FGFR is. And I on purpose put them as like two Y's because, as we know, they can fuse together, and that's really what we are after in regard to illness. And by the way, they're not really specific to cholangiocarcinoma. They happen in many illnesses. And if anything, not only in cancers. Some of you might know about FGFR, and this is an important example of Apert syndrome, which is related to FGFR. And sadly, and I chose it for no reason except it illustrates that fusion in a graphic form, even though, of course, it's a totally different thing. But this is how children are born with actually fused appendages or or fingers, and they need splitting apart, hopefully until we get some genetic alteration for that disease as well. With this said, in cholangiocarcinoma, proudly with collaboration for many colleagues who are in the meeting today, many phase 2 studies evolved, among which the first one, which our dear colleague, Dr. Javli, presented on behalf of all of us on many occasions, was in JCO and then showed up in the abstract format at the GI ASCO this year. And if anything, it showed a clear improvement in regard to progression-free survival and overall survival. Understandably, this is in comparison to the standard control that we know about from chemotherapeutic range. This is updated data with 12.2 months, and we're waiting to see where this might evolve further. But along that line, another one, which is pemigatinib, which we proudly led in collaboration again with many colleagues, among which is Dr. Murad. And we can see over here that we went after allocation for the translocation for other alterations, and of course, without alterations. And with this, we're able to find a pretty drastic response rate of 35%, including some CRs. And add to this, we showed an improvement in survival, especially among those with diffusion, up to 22 months. Actually, this effort led to the approval of pemigatinib by the FDA for patients with FGFR2 genetic alterations, because even though to a lesser extent, but it happens in other alterations as well, as you can see from cohort B and C, but more importantly, it was approved in second-line setting. Please keep this in mind. We're going to talk about it at the end of the presentation. Other efforts, which again to give credit for many colleagues as well. One of them is futibatinib, which, as you can see over here, again, a pretty robust response. I would nominate that this is really a response that is probably clearly showing a certain stability of disease. But if you are to really focus on the blue in color parts of the waterfall plot, you'll see that quite a bit of them, especially in the response, are cholangiocarcinoma. And there's definitely something evolving still in the works. Derazantinib is another one. I don't need to keep repeating the great responses that we're seeing over here, and you can see quite a bit of PRs. Interestingly, derazantinib is being looked at, or a certain curiosity brought in on certain mechanism action that it might be looked at into mutations, and already a study is ongoing with patients with mutations of FGFR with the treatment with darzanetinib. And of course, DB1347, again, one more time, another alteration of FGFR2 that led to responses, as we can see over here. We don't know where it's going to evolve further, but I think it gives you a taste about, like, yes, there's a lot of interest and good for the patients in regard to the FGFR2 genetic alterations. Resistance can happen. I give credit for our dear friend, Dr. Goyal from MGH, who really had this seminal paper. And we're pretty much in really quite laborious work looking at specific tumors in the liver, taking the actual biopsies on them even at autopsy, and this is very important, and try to find out if there's genetic alteration. You can see, yes, there was genetic alteration that was being depicted, and among which is the gatekeeper, the FGFR2 V564F. The question that I bring up here, do we need further work on that? Yes, this is more descriptive, but definitely more interaction and more evaluation of that alteration will be key to happen. And the question is it something that's happening by random and just happen because the tumor is under stressor of a certain anti-FGFR2, or is it something that can happen because of chirality of a chaos? This is not to depict anything that is related in any way, shape, or form regarding to the FGFR2 fusion. But interestingly, we know very well nowadays that all those fibrils that we have in our bodies are actually based on a certain chirality that will kind of be mathematically calculated to really bring in that what we call in mathematics chaotic phenomenon. And the theory that we're bringing up here, and Dr. Goyal and myself and others, including Nabil Bardisi, we're at least discussing at this point in time, hoping that we'll be able to delineate it further and find out if that kind of spirality add to the fusion will be able to delineate exactly what triggers those stressors and how do they evolve into that specific alteration that, of course, if we're to predict it, we'll know very well at least what to do rather than wait for the unknown or the unexpected. I will leave you with the last question, which is, where do you go from here? This is great data. We have only one conditional approval. Where would you go from there? As we all know, we have quite a bit of efforts that are going on because we are all interested in what we call overall survival. And this is really what the racetrack should be looking at in regard to overall survival. And interestingly, though, we know very well that now with many therapies are going, survival might be a little bit of a challenge. These are at least 3 of the studies currently ongoing in regard to phase 3 clinical trial looking into figratinib, which I already presented, which was BGJ398 to begin with, pemigatinib, which I also presented the phase 2, and the futibatinib, which I know Dr. Goyal and many others are involved in. Intriguingly, these studies, this is all published data from the clinicaltrials.gov, have already started accrual, at least in December 2019, the least, and March 2020. So at least they are in the year plus in regard to accrual. If you were to really do guesstimate work of math about where do they stand in that regard, this is really how much is going to take time for when these studies are going to finish. About 40 years. I can assure you, I will not be here. I'll be sitting on the beach. I don't know about you, but I will not be here. And the question is, more importantly, can our patients wait that long to get those answers? And if anything, I would say that rather than doing those kind of efforts in the standard setting that we all know about in regard to the overall survival, the question is, can we depend on something else? Because if anything, there is a certain effort currently at least being looked at. This is not yet reported, but many of us know about it, that we're trying to join all those 3 in one joint study that will have 4 arms, 1 for each, and compared to the standard of care, because they're all compared to standard of care. But even if we were to do that, and if anything, we're building a study, let's say guesstimate work, and I'm not revealing any details because the details are still in the works, but guesstimate work, if you end up like, let's say as guess 100 patients each arm of those, you still need at least 30 to 40 years to finish the study. And the question is then, instead of depending on the overall survival, can we depend on the relay perspective? Because after all, rather than looking at progression or at overall survival, it's our obligation, all of us, to talk to the agency and relay about that a progression-free survival, duration of response could be very important indicatives of value. Same way pemigatinib was approved, why not infigratinib and many others will be approved the same way? This is really where the effort is going to be, rather than dependent on overall survival studies that are very old with us. They've been with us for the last at least 50 years. And in that specific setting, we're talking about a rare cancer, cholangiocarcinoma, which is a great credit to the foundation taking care of the patients with cholangiocarcinoma and be their advocates. And number two is with a genetic alteration that's really rare. I know we all quote data about 15%, 20%. I tell you, in reality, if we're lucky, it's 5%. And that's why a PFS or duration of response phase 2 trial should be more than needed for the purpose of the approval of those drugs, rather than wait for those elaborate, long, and pretty involved studies, or even join them all in one study, because it will take the same time and will really, we will be missing on the point and missing on the opportunity that we can provide for the patients. And that's why progression-free survival duration of response will be important, because we have many runners on the field, among which GEMCIS, that Dr. Valley provided with his colleagues from the UK. We have the FOLFOX6, Dr. Lamarca and colleagues were also provided. And of course, the FGFR2 will come and play as well in that regard. So with this, I will go back to the pemigatinib, and I'll just give it as an example. Is this satisfactory enough for us to really approve the drug? I personally, yes, it is. And I'll be eager to hear from all of you about your thoughts when we come to the discussion. Again, I thank you for having for the invitation, and congratulations on a great meeting. Thank you very much.